Good afternoon, everyone, and welcome back to Science in the Age of COVID-19. Today, we are extremely excited to welcome Aashish Manglik from the School of Pharmacy at UC San Francisco. Aashish has, like many other scientists in this pandemic, completely changed up his research program to deal with the pandemic and try to help us get it under control. What he was doing before COVID was his lab was very influential in the study of various transmembrane proteins, usually G protein coupled receptors, particularly opioid receptors, which help in the perception of pain and other signaling events. And then when COVID hit, his lab completely repurposed itself to, they are involved in figuring out what are the best ways to test for the existence of antibodies in patients to see if you've been exposed to SARS-CoV-2. And then what his lab has done that is extremely important is they screened for binders to the SARS-CoV-2 spike protein and came up with one that is extremely high affinity and is in such a format that it will be a very promising drug candidate moving forward. So take it away, Aashish.
Thanks, Loren. And I'm excited to be, part of this series. I must admit, I feel a little bit intimidated, with the remarkable people, true virologists who've spoken before me. And, I should preface this talk with, I know very little about virology and have tried to catch up to the small extent possible. But my lab normally works in a completely different area, as Loren mentioned. And the project that I'll tell you about today was really a chance to meet the moment, really driven by an amazing set of trainees here at UCSF. So the premise, again, is laid out by this title slide, which is our development of aerosolized nanobodies for SARS-CoV-2 passive immunization. But just to give you a sense for what we normally care about, our normal research program is very interested in how cells sense and perceive the outside world. And it turns out that the professional sensors that enable cells to do this are the so-called G protein coupled receptors that sit on the surface of our cells. So these remarkable proteins enable our bodies and our cells to sense everything from hormones, proteins, ions, pH, light, or even force itself. And the common structure of these proteins then enables these incredibly varied stimuli to then engage a whole host of processes inside the cell. So normally our research program is very interested in the chemical logic that enables our cells to sense these stimuli. And one of the technologies that we've been developing over the years are ways of pushing and pulling on these receptors to make them amenable for structural biology studies, or eventually to bridge what has become an enormous revolution in structural biology with really their cellular biology. So our long-term goal is not just to understand what these proteins look like at the atomic level when they're doing their business, but really where do these conformations exist in cells and even in tissues and animals. So one of the technologies to get at this that we've been developing are so-called conformationally selective nanobodies. The idea is as such. Let's say you have a membrane protein, for example, a G protein coupled receptor, and a lot of work over many labs over the past 2 decades has revealed that these receptors aren't just simple on-off switches. They're really complex. They exist in many different shapes. And what we try to do is raise single domain antibodies, or so-called nanobodies, that recognize one specific shape or one specific conformation of these receptors that's important for their signaling. So for example, this nanobody, binding to this cartoon receptor would only bind to the active conformation that's able to do its signaling process. We've shown that we can find nanobodies like this for a whole host of G protein coupled receptors. And in very recent work, we've used library-based approaches to find nanobodies that, for example, in this case, bind to a receptor that transduces the hedgehog signal. This is a receptor called smoothened. And just as an example, this is the nanobody that we raised that binds to smoothened only in the active conformation and then allows it to basically sense whether this receptor is active. We've then try to establish use of these nanobody reagents to go sniff for active conformations of these receptors inside cells. So in a simple experiment, we can hook up one of these nanobodies to a GFP protein and then express that construct in cells expressing the receptor, and then watch, for example, recruitment of that nanobody to the plasma membrane when the receptor becomes activated. This is work in collaboration with Mark von Zastor here at UCSF, who really pioneered this approach. Now, one of the big bottlenecks to identifying nanobodies for academic labs across the world is the ability to have access to llamas or alpacas where these reagents normally exist. And working with Andy Cruz's lab at Harvard, we've developed synthetic libraries where instead of having to go immunize an animal, you can use a yeast surface displayed library of nanobodies to rapidly go find nanobody against any given antigen. And some of the previous data that I showed was really was enabled by some of these technologies. So that's again our normal research program. When the pandemic hit, a really remarkable group of trainees, primarily led by Michael Schuff, a graduate student in Peter Walters' lab, decided to use some of these technologies that we've been developing to try to tackle the pandemic. And he was joined by, again, a cadre of graduate students and postdoctoral fellows in my lab. Brian Faust, for example, really did a lot of the structural biology work that I'll show, along with other collaborators here at UCSF and across the world to understand whether our reagents might actually work against the actual virus. And then a lot of the structural biology work that I'll show you coming up was done by a really new way of doing structural biology that was led by Oren Rosenberg and Klim Verba. Okay, so what's the premise of the project? So this is probably now familiar to most of the watchers of this series, but I want to highlight that the fundamental way that enables the coronavirus, or any coronavirus, but in particular SARS-CoV-2, to enter cells is a binary interaction between the spike protein on the surface of the virus and the angiotensin converting enzyme 2 receptor for the virus on host cells. And this binary interaction is absolutely critical for the virus getting into cells in the first place, and is a rate determining, or the critical step that leads to infection. If we zoom in on that, on this binary complex, this is beautiful work by many in the field prior to even the current pandemic that have mapped this interaction between spike and the ACE2 receptor. So blocking this interaction is a very potent way to neutralize viral entry. And as a result, a number of the antibody strategies, both from the perspective of trying to coax your own immune system to develop antibodies, in the context of vaccines, or by delivering antibodies exogenously, aim to block this interaction to prevent infection. So there's two possible ways of providing passive immunity. And again, here, passive immunity is distinguished from active immunity from the notion that instead of coaxing your own immune system to generate antibodies, perhaps you can give antibodies that block that interaction and provide some benefit. Now, one of the tried and true methods, or one of the oldest methods for providing passive immunity, is to use convalescent plasma. Sorry, I misspoke. Tried and true is a little bit too far, but it's an old idea. The idea of convalescent plasma goes back about 100 years. The idea is simple. If you have any new pathogen that infects a person, and you can identify patients who've recovered from that disease, presumably they've made antibodies that neutralize that virus or that pathogen. So if you can identify those individuals, collect their blood, and in this case, the modern era, collect the portion of blood that contains antibodies, in this case plasma, you could give that to patients that are sick with the disease, and that would then transfer some of these neutralizing antibodies and potentially provide benefit. Now, as many of you are probably aware, the FDA provided an emergency use authorization in late August for hospitalized patients with COVID-19. And very immediately thereafter, in September, an NIH panel found that there was insufficient data to really recommend the use of convalescent plasma. Really, the big challenge for this approach, while it's exciting to see that this approach could be essentially applied for any new pandemic, the challenge is that the safety is unclear, and there are not really standardized protocols for determining the titer of patients that that would be good donors, and how to match them up with ideal patients. So really what we need are prospective randomized trials to make this approach work. Now, in contrast to convalescent plasma, a more recent approach to providing passive immunity is the use of monoclonal antibodies. And the idea here is that instead of just giving the bulk set of antibodies that a patient, who's recovered from the virus, but instead, we try to identify a single antibody that perhaps has really remarkable properties. And once we can identify such a neutralizing antibody, either from some sort of biotechnological approach or really from working up individual antibodies from recovered patients, that could be manufactured at scale and given to patients that are sick with the coronavirus. Now, there's multiple candidates in clinical trials, and for those of you who are following the COVID infection for our president, this is one of the experimental approaches that's been given to him. That's a Regeneron cocktail. But really, fundamentally, monoclonal antibodies for passive immunity require intravenous dosing, either for treatment or for prophylactic use. And for prophylactic use, one of the big challenges is that the amount of doses required are really quite high, something on the order of 50 milligrams per kilogram. So if you actually look at the dosing of some of these antibodies, they're on the order of gram scale per person. So that combined with the relative expensive production of antibodies means that while these approaches are likely going to have some benefit in the treatment of patients who are acutely sick, truly prophylactic use, for widespread deployment is relatively unlikely, just because of the dosing required and the relative expense of production of these molecules. So, this is a lot of these ideas about monoclonal antibodies were already being, many of those programs had already gotten off the ground by the time we started our project in March. But we considered perhaps taking a different approach. So instead of perhaps providing passive immunity via the intravenous route, we wondered whether it would be possible to provide passive immunity by either delivering molecules that would neutralize that interaction with spike, either through an aerosol, through one of these little nebulizers, or perhaps even simply through a nasal spray similar to an Afrin or a Flonase, if any of you've used those. Now, the main advantage of this approach would be that it could be self-administered and it wouldn't have to be given through the blood. And just from first principles, this may enable patients who would otherwise not need to be in the hospital to actually have some way of tackling illness. And because an approach like this would be delivered directly to the site of early infection, the dosing required for tackling the virus may be lower. Now, this doesn't come without any challenges. The first challenge to really be able to achieve something like this is that, aerosolizing a protein or putting in a little nasal spray requires the protein molecule itself to be ultra stable. But on top of that, there's really a relatively, there's no approved approach or FDA approved approach to doing this. So really the pharmacokinetics or a lot of the other clinical development approaches are not completely clear. Now, in order to do this, what we decided to use again was our technology to develop nanobodies. But let me give you a little bit more background about nanobodies versus monoclonal antibodies. So this is the structure of a conventional antibody that's flowing in my blood or your blood. And it's a relatively complicated molecule. It's heterodimeric, so it's composed of a heavy and a light chain. And it has a constant region that has its own functions in revving up the immune system. Now, this is the business end of an antibody, and this is what enables antibodies to bind almost any given antigen. Now, it turns out that llamas and alpacas and camelids make so-called nanobodies, and these little proteins are just derived from the variable region of the heavy chain. But what's unique about nanobodies is that they're very small, about a tenth the size of an overall antibody. They're single chain proteins. They're ultra stable, and even if they denature, they can refold. They're not glycosylated, and they're similar enough to human antibody heavy chains to not lead to an immunogenic response even in patients. And finally, because of their relative simplicity and their stability, they're relatively easy to produce in microorganisms like yeast or even in bacteria. So is there any precedent for delivering a nanobody to the airways for any respiratory virus? And it turns out that a company called Ablinks had been trying this approach for a different virus called respiratory syncytial virus. Now, RSV is a virus that that infects adults and children, but primarily brings infants into the hospital if they have a particularly bad infection with this virus. So Ablance in the past had developed a trimeric nanobody, so 3 individual nanobodies linked together with a little protein linker that would bind to the fusion protein analogous to the spike protein of SARS viruses. And they were able to show that this trimeric nanobody very potently neutralized RSV. But importantly, on top of that, this molecule ALX0171 was stable to aerosolization. So this is a size exclusion chromatogram showing that even if you aerosolize this molecule, there's very little presence of high molecular weight aggregates. And then they went on to really develop a lot of the models to see whether this might be effective. So in a lamb model of infection, they could show that this molecule would prevent viral loads after treatment. And in healthy human adults, they established that the half-life, if one were to deliver a molecule like this to the lungs, would be about 20 hours, suggesting once a day dosing. But only very recently, only in the past about week or so, have they released data from a phase 2 trial where they treated infants with RSV, that were infected with RSV with this molecule. And this is the data from this very recent publication. So I'll just walk you through this data because I think it's pretty exciting in some ways, and then I'll give you the kind of downside afterwards. So what you're looking at here is hours of treatment on the x-axis, and then just simple viral plaque assay to see whether the virus can be detected in the nasal turbinates of these infants. So placebo group, you can see, clears the virus relatively slowly. Takes a couple of days after infants present to the hospital for the virus to clear. But by contrast, the highest dose of this molecule, ALX0171, even after a single dose, in about 90% of these infants, completely prevents viral replication. And you can see that, basically after that first dose, almost 90% of patients don't have any detectable virus. Now, the downside of this study was that the molecule, although very effective at preventing infections or preventing viral replication, didn't really improve the clinical severity of these infants infected with the virus. And we think that largely reflects the fact that infants that come to the hospital with this specific virus present relatively late in the course of infection. So really the benefit of these approaches of preventing viral entry largely is probably for patients that are relatively early in the course of infection. And as a result, probably this specific indication was not ideal. But just the fact that this approach of delivering a biologic through the airways to limit viral replication is really quite exciting to us. Okay, so let me tell you a little bit about what we ended up doing. So using this platform that we've developed in my lab to identify nanobodies rapidly, we identified a number of nanobodies. So in my lab, what we do is we take inspiration from individual llamas or alpaca, but then develop synthetic libraries, and our latest libraries now are about 2 billion variants, where each individual nanobody sequence is, again, tries to recapitulate the kinds of nanobodies seen in animals. And then we display these nanobodies on the surface of yeast. So the idea here is that we have 2 billion individual sequences with each unique sequence displayed on the surface of an individual yeast cell. And then what we can do is use this library to go find binders to any given antigen. And this is the kind, this is what we were doing before the pandemic to find nanobodies against G protein coupled receptors. But in this instance, we used this library to find nanobodies that would bind to the spike protein. So this is now purified spike protein labeled with, for example, a fluorophore. Then we could select binders, discard the clones that don't bind, and then iterate that process. And what we ended up with were about 21 nanobodies that bound to spike. We selected the ones that were the most stable for further optimization. We improved one of those on its own to bind with picomolar affinity to spike. And then using structure-based approaches, we engineered a construct that binds to spike with femtomolar potency. And then finally, we humanized our molecule, or basically converted its sequence to be as human as possible to decrease the probability of an immunogenic response. So I'll walk you through some of the specifics of what we actually did. Now, very early in the pandemic, we were enabled by the remarkable approach to sharing reagents and knowledge that's become relatively commonplace now, for at least for coronavirus research. And we were able to obtain a construct, a pre-fusion spike construct from Jason McClellan's lab. And we really targeted using the full-length spike protein, or the full, at least the ectodomain of spike, instead of just the isolated receptor binding domain, to find nanobodies that would inhibit the spike ACE2 interaction. Our hope was that we might not just find nanobodies that block this interaction, but perhaps again find nanobodies that do this via unique conformational mechanisms. These are just, again, a simple size exclusion chromatogram showing some of our preparations in a simple SDS page gel. And again, early on, we could show by size exclusion chromatography that our purified spike protein, for example, labeled with an Alexa dye, could bind purified ACE2. And that affinity we could validate, again, work that had been done by others prior to us to be in the kind of double-digit nanomolar range. With Yifan Cheng's lab here at UCSF, we were able to boot up our EM microscopes almost a couple of weeks right after UCSF entered our shutdown, and determine a pretty reasonably nice cryo-EM structure of just that apo spike protein. So we were pretty confident that the material that we were making here at UCSF was analogous to the material that Jason's lab had been producing and had previously solved the structure of. So with this reagent in hand, we went into our library to find nanobodies that bind spike. So this is the general idea. We start with our library of 2 billion variants, and over progressive rounds of selection, try to find clones that bind to spike with higher and higher affinity. And then finally, we looked for clones that would bind to spike, but were blocked for binding by ACE2, by purified ACE2 itself. And that's how we ended up with 21 clones. Now, when we looked at these nanobodies, they fell into two classes. And I'll walk you through the data here because I think we found a very unique class of nanobodies that probably have a very interesting mechanism of action that we don't completely understand. So on top are flow diagrams, flow cytometry diagrams for one class of nanobody. This is nanobody 6. This is probably the easier one to understand. And on bottom is that class of nanobodies that we don't quite understand how they work precisely, but they have really interesting properties. So in each one of these flow plots, what you're looking at is a clonal yeast now. So all of the yeast on the top are expressing only nanobody 6 on their surface. On the x-axis is whether these yeast bind to purified spike protein labeled with an Alexa 647 dye. And on the y-axis is whether the nanobody is even on the surface. So the fact that there are yeast now in this double positive gate tells you that the yeast with nanobody 6 on their surface are able to bind to the spike protein. If we repeat that experiment in the presence of ACE2, all of that binding goes away. So this really clearly suggests to us that these nanobodies bind to an epitope on spike that's the same as ACE2. Now, if we do that same staining experiment with just the isolated receptor binding domain of the spike protein, we see, again, pretty robust binding, suggesting that this kind of nanobody binds to the receptor binding domain in a very similar epitope where ACE2 binds. The nanobody on the bottom is pretty unique. So again, it binds to the spike protein. But if we add now ACE2, there's a little bit of a decreased binding, but not complete. And if we repeat that experiment with just the isolated receptor binding domain, we see very little binding. Now, this suggests that this kind of nanobody binds somewhere else on spike and is able to moderately decrease binding of ACE2. So these are probably allosteric molecules or act by some other mechanism of action to disrupt interactions between spike and ACE2. So at this point, we had a number of nanobodies in hand, and we wanted to characterize them pretty rapidly. And typically for most structural biology projects, you'd really whittle it down to one clone that you really liked and then go to the ends of the earth to find out really how it works by structural biology. And here we were incredibly enabled by a new way of doing structural biology, which is a group that we call the UCSF Coronavirus Structural Biology Consortium, led here by Oren Rosenberg and Clem Verba, along with 18 other UCSF principal investigators and a team of about 60 trainees, both graduate students and postdocs. And this group has basically created essentially a pipeline approach to being able to get structural biology of coronavirus-related proteins pretty rapidly. And these are some of our EPIC Zoom meetings to try to coordinate these efforts. So the idea here is that unlike what we typically do for structural biology, where it's one lab really doing everything from the very early work to the very late work of a project, we've split the work such that individual faculty lead essentially a part of a pipeline to basically enable this to go a lot faster. So with this kind of a group, at our backs, we were able to get structures of many of our nanobodies bound to the SARS-CoV-2 spike protein. I'll tell you about two of them because they're particularly illustrative. So we were able to solve the structure of spike bound to one of our lead nanobodies, which we call nanobody 6, both in two conformations of spike. So both in spike, where it's completely closed with all of its receptor binding domains laying down, as well as a lower resolution structure of spike bound to nanobody 6, where one of the receptor binding domains has flipped up and could engage ACE2. Here, again, you're looking at it from the side on these top panels. Nanobody 6 is in red and spike is in blue. Looking top down, you can see, again, the nanobodies in red and spikes in blue. We were also able to determine structures of another nanobody called nanobody 11 that binds, again, to spike in the both closed state with all the receptor binding domains down, or in the open state with one receptor binding domain flipped up. Now, the way in which nanobody 6 engages spike is really quite unique. So I'll talk about nanobody 11 first, because I think that's more indicative of how many other nanobodies or antibodies bind to the spike protein. So the way that nanobody 11 binds to spike, so for example here, this nanobody 11 binds to this receptor binding domain of spike. And on the right panel here, I'm just showing a kind of a footprint of each one of these nanobodies compared with ACE2. And for example, what you could hopefully see is that ACE2 has this kind of footprint on this receptor binding domain, and nanobody 11 simply competes with that same epitope and thereby prevents ACE2 binding. Now, what's unique about nanobody 6 is that it again also sits down on this gray colored receptor binding domain to block ACE2 binding. But on top of that, it has one of its complementarity determining regions, its CDR3, reaches over to a neighboring receptor binding domain and makes interactions with that receptor binding domain in its down state. So what happens as a result is that a single nanobody 6 binding to this receptor binding domain not only prevents ACE2 binding by sterically competing, but by locking the entirety of spike into the receptor binding domain down state that's incapable of interacting with ACE2 in the first place. So this dual mechanism we think is very unique. And just a very recent paper by David Wiesler's group shows a very unique antibody that a regular, conventional antibody that perhaps functions the same way. So we think this dual mechanism may be kind of a preferred mechanism if achievable. Now, the fact that this nanobody, that nanobody 6 binds in this very unique geometry enabled us to design multivalent versions that we predicted would have incredibly high potency. So one thing that we could do with these structures is just map out the distance between the C terminus of one of these nanobodies and the N terminus of the neighboring nanobody. And for nanobody 6 bound to the closed conformation of spike, that distance is only 52 angstroms. By contrast, that distance is 72 angstroms if nanobody 6 is bound to open spike with one of these receptor binding domains in the up state. And for nanobody 11, that distance is on the order of 70 angstroms. Now, we could design linkers that would span this distance, and that's of about 15 or 20 amino acids, but that distance, that linker would not be long enough to span 70 or so angstroms. So here's what happened when we designed these multivalent reagents. These are Biacore or surface plasmon resonance experiments. What you're looking at here is the experiment is that we lay down spike or purified, just the ectodomain of spike in this prefusion conformation on the chip. And then we're flowing over either nanobody 6 or a bivalent version of nanobody 6 or a trivalent version of nanobody 6. So you can see nanobody 6 on its own binds very rapidly and then falls off very rapidly. The half-life of dissociation here is about 12 seconds. If we make a bivalent version of nanobody 6, there are now two phases of dissociation, a fast phase and then a very slow phase. We think the fast phase represents if one domain from this bivalent antibody 6 engages with an upstate RBD, then that can fall off kind of just the monovalent dissociation rate, and that's basically, you can see this dissociation rate. The fast phase is pretty similar to the dissociation rate of just a single monomeric nanobody binding. But then the slow phase here reflects two nanobodies locking down on adjacent receptor binding domains. And this slower dissociation phase now has a half-life of about 37 minutes. If we now look at the trivalent nanobody, the slow phase is immeasurably slow. And in fact, we can bound it, but really, it's too slow for us to really get an accurate measurement on. Now, we've been presuming that this biphasic dissociation reflects, again, the up versus down motion of the receptor binding domain. So we tried a simple experiment. If it's true that that's what we're seeing in the fast, in this biphasic dissociation, then simply changing this association period should influence how much of the spike is bound in that triple engaged format. And when we do that experiment, if we change the association time from 4 seconds to 520 seconds, you can see as we wait longer and longer, more of that spike has a chance to basically go into the all down state where all three of those antibodies are engaged. And as a result, once that happens, we observe basically, again, only the slow dissociation phase. That happens with a half-life of 65 seconds. So we think this really reflects the fact that this trimeric construct can engage all three of these receptor binding domains in this down state at the same time, really locking spike in this inactive conformation. So with those biochemical and biophysical assays in hand, we turned to trying to understand what multivalency buys us for viral neutralization. So here are data now in a pseudovirus assay, where we're looking for neutralization of pseudovirus infecting ACE2 positive HEK cells. And you can see that the parent nanobody 6 here inhibits with the micromolar affinity, but that's simply making that trimeric version has a profound gain in affinity of potency of almost 4 orders of magnitude. By contrast, this other nanobody, which I showed you was too far away to basically engage multiple receptor binding domains in that trimeric construct, has a gain in affinity, but it's nowhere near as large as what we see for nanobody 6. Again, reflecting that the way that nanobody 6 binds enables us to create constructs that can engage all three of those receptor binding domains at the same time. What was even more impressive is when we looked at this against live SARS-CoV-2, this is work in Marco Vignuzzi's lab, the potency gain was even more remarkable, and this trimeric form of nanobody 6 inhibits viral entry with picomolar potency, about 30 or so picomolar, if I remember correctly. Okay, so that's where we were. So we had identified nanobody 6, or what we call now AERONAB 6, and trimerized it, and that's the data that I've shown you. But then we took 2 additional strategies to make a better set of binders. One was to humanize the nanobody. Again, these nanobodies that come out of our library are really designed based off of kind of a core scaffold that comes out of llamas. And we wanted to make sure that to decrease any probability of an immunogenic response, to remove the kinds of differences between human gene and the llama gene. So we made a humanized version of it, and separately, we wanted to further increase the affinity of nanobody 6. So we affinity matured it, and then we combined those two to make a trimeric form. So this thing in the red here on the bottom, a higher affinity version and a humanized version and a trimeric version of that is what we're hoping to take forward. So in order to make a higher affinity version of just that monomeric nanobody, what we did is created a new library. We took nanobody 6 and then created a saturation mutagenesis library where we vary every single position on the complementarity determining regions. And then we did 2 rounds of selection and identified 2 mutations that were highly penetrant in a population of clones that were binding with higher affinity. So I showed you the previous, if you recall the previous data, where the parent nanobody 6 binds very rapidly and comes off very rapidly. This affinity matured version of nanobody 6 again, binds rapidly, but on its own dissociates really quite slowly, on the order of with a half-life of about 30 or 40 minutes. And then when we make a trimeric version of that nanobody, it binds and basically now never lets go. So it again has a dissociation rate that we can bound that suggests sub-picomolar potency. But again, these are estimates because just the chemistry to link spike onto the chip is now limiting to get accurate measurements. So again, really a pretty highly potent molecule when it's in its trimeric format. We were able to determine a structure of spike, again, in its closed conformation bound to this high affinity nanobody 6 or matured nanobody 6, and that's shown again in pink here. And then we can start to compare, for example, what are the reasons why we get this really remarkable gain of potency of about 500-fold. So like the parent nanobody in red here, the matured form largely binds in a very similar way, again, engaging both a primary receptor binding domain to block ACE2, but also engaging a neighboring receptor binding domain to lock spike in this inactive conformation. And what was remarkable here is that while one of these mutations optimized kind of the local geometry, so the parent nanobody in this case has an isoleucine at this position, and the affinity matured variant has a tyrosine, which you could perhaps see kind of fits in this little hydrophobic pocket. It makes a hydrogen bond. Another mutation in CDR3 had a pretty profound change in the conformation of the loop. So the parent nanobody in red here has a proline at this position, and substitution to a tyrosine causes this loop to change pretty dramatically in conformation, which is something we relatively rarely observe in antibody affinity maturation campaigns. We were also able to get a structure of this nanobody, mature nanobody 6, on its own. And when we did that, we were pretty surprised to see that the conformation of these loops is really quite different between the nanobody bound to spike and unbound to spike. So in green here is a crystal structure of this nanobody on its own. And in pink, again, is the structure bound to the spike protein. And what you can hopefully see is that these loops are in completely different conformations when they're bound to spike or not. And this is, again, something we don't observe all that often, or when we look at antibodies in the unbound or bound state. And we think that these conformational differences may be what underlie the remarkable gains in potency that we observed of about 500-fold going from the parent nanobody to this affinity matured version. Now, the last thing that we wanted to do was to humanize this clone. So again, our library is designed to really recapitulate the diversity of sequences found in llamas. So the core scaffold on which our nanobodies are built is a true llama scaffold, on which there are these three complementarity determining regions. This is, again, the data that I've already showed you on the left here, Biacore data for our trimeric affinity matured nanobody 6. And what we next wanted to do is transplant these three loops into a human gene. In this case, one of the closest human heavy chain genes is IGHV3-66 to the llama scaffold, while introducing 5 mutations to still allow this little human gene to remain soluble. And with a little bit of engineering, we were able to finally find a construct that is now largely human, with these loops that bind to the spike protein with potency that's basically unchanged compared to the llama molecule. So this is, again, this is data now for the humanized version of this nanobody, which binds with very similar potency to the true llama, to what came out of our library and was optimized. So that's where we are now. We have a molecule that binds with very high potency. That's this trimeric nanobody on a core human scaffold. So again, with Marco Vignuzzi's lab, we've been able to test whether these molecules are effective against the authentic SARS coronavirus. And here's kind of the progression of our molecules. This is, again, data looking at infection of SARS virus into Vero E6 cells. And the initial hit that came out of our library, which is nanobody 6 or AeroNab 6, inhibits with not particularly remarkable potency of about 5 micromolar. But simply making a trimeric version of that gave us a really remarkable boost in potency. And again, we think that this remarkable boost in potency reflects the fact that this specific nanobody can be designed to engage 3 receptor binding domains at once. So here again is in dash blue is that parent nanobody, and that trimeric one, allows us to gain almost 4 or 5 log orders of potency in this assay. Maturing or affinity maturing that individual monomer allowed us to gain something like 2 or 3 log orders, again, going from this dash blue line to this matured line in green, and the dash green line. And then combining the multivalency strategy with the affinity maturation gave us a clone that inhibits now really quite potently in the 30 picomolar range, which is among the most potent, antibody or nanobody-based neutralizers that's been described in the literature. And then finally, in red here is the humanized molecule, and you can see that it basically loses very little in potency. And this is really the molecule that we think may be amenable to bringing to patients. Okay, so one of the premises of this project is not just to develop very potent molecules, but molecules that may be stable enough to deliver via an aerosol. So some thermostability experiments that we did with these experiments have revealed that they have melting temperatures that are in the 60-degree range, which, while not being atypically high for antibodies, is certainly in the range that may enable us to aerosolize them. But really, evidence for aerosolization we've been able to gather is pretty exciting. So this is a little cheap little aerosolizer or nebulizer you can buy on Amazon. Let's play a little video of that. And what we were able to do is load up the top of this little nebulizer with some of our nanobodies, aerosolize it, and then collect this aerosol to see whether our nanobodies, basically remain stable or effective or retain their function after this procedure. And what we were able to show by size exclusion chromatography first is that this trimeric nanobody doesn't aggregate upon aerosolization. And on top of that, if we try to lyophilize it, we also don't observe any aggregation. More recently, we've been able to show that that aerosolization, lyophilization, or treating these molecules at 50 degrees for 1 hour doesn't cause any loss in function. And these are now pseudovirus assays showing the nanobody on its own in purple. And then if we treat them with these various conditions, we again observe no loss in potency, suggesting that, again, they're pretty robust proteins that may enable aerosol-mediated delivery. So with that, we think we have molecules that may enable this alternative approach to passive immunity, either delivered through an aerosol or perhaps as an intranasal spray. Again, the main advantage of an approach like this is that it could be self-administered. And if you think about, what we do for patients that are pretty early in the course of infection, perhaps not sick enough to go to the hospital, right now, the main thing that we tell them is to basically, stay at home and quarantine from others. And we think that for those patients or, household contacts or other individuals who've just learned that they were, exposed to someone that has coronavirus, we think an approach like this that's targeting very early phases of infection may have some great utility, especially because it can be self-administered and doesn't require patients to come into the hospital. Where we are now is really trying to understand, what are the challenges required to get this into patients, and that's where we are, with the kind of classic academic translational valley of death. So this is a picture that I took with my wife when we were returning from Las Vegas coming back to California. This is Death Valley. And, we're probably somewhere kind of in the middle of entering this valley, and we're hoping that we can find a path forward to provide this kind of very unique approach to tackling the pandemic. And even if it's, not here in time for this specific pandemic, we think this kind of approach may have great utility for other pandemics or other respiratory viral diseases. So with that, again, I'll thank the people, a really remarkable group of trainees in collaboration with Peter Walter's lab here at UCSF, that have really driven this project forward in pretty excruciatingly challenging times, with work restrictions. A number of groups here at UCSF and across the world that have enabled us to understand how these molecules work and whether they work against the real virus, as well as this remarkable consortium of structural biologists that have enabled us to do structure-based design really in real time, which has been pretty exciting. So with that, I'll stop and take questions. And yeah.
Thank you, Aashish. That was a very engaging talk. And yes, we have questions. I'm going to call on Evangel. Would you like to ask your question? Okay, I'm going to read out her question. What is the reason for leaving the humanized part to the very end?
Yeah, that's a great question. So why not just build a library? I think I'm presuming the question is, why not just build a humanized library?
Yes.
Yeah, this is mostly for historic reasons. One of the things that we have, for most of our work that has nothing to do with trying to develop drugs or develop molecules, we've developed libraries that are llama based, primarily because one of the longer term goals in the lab is to try to understand how camelids build happy nanobodies or nanobodies that are highly stable. And we don't want to stray too far from what's seen in natural immune repertoires to try to learn those principles. We are, this is a point well taken. We are right now building libraries that are based on humanized scaffolds, but just historically that hasn't been a priority because we're not, normally in the business of trying to make molecules for human use.
Yeah, I think I was in particular thinking about, not about the starting library, but you go also through a process of optimizing based on the structure and other things. Why not do it in the context of something that has already been humanized? I don't know.
Yeah. This, I think we would, just practically how this project went was to take these parallel approaches at once and then try to combine them at the end instead of, doing them a little bit more in a linear fashion. So I think typically what we would do, would be to take hits from the library and then maybe humanize them and then maybe, affinity mature the humanized molecule. And just in the context of the pandemic, to try to move fast, we just, basically threw everything in parallel and then hope that, the combinations would work and remarkably luckily in some way that they did. You could imagine that those individual strategies, may not have worked together, but our hope was just that they would and we were able to, save months in the process of by taking that parallel strategy.
Yeah, spectacular. Thank you so much.
Thank you, Aashish. Our next question is from Brian. Brian, please unmute yourself. I've actually unmuted you. Go ahead.
Can you hear me?
Yes.
Hi, Aashish. This is Brian Roth. How are you?
Hey, Brian. Good to hear your voice. Yeah.
Amazing technology. So you may have mentioned this, but I didn't hear it. So I guess the question for aerosolized nanobodies is their physical chemical properties. Do they stay aerosolized all the way down to the sort of terminal alveoli? Have you been able to determine that? I guess because that would be probably the therapeutic point of contact. Yes.
Yeah, this is a great point, Brian. And here again, we're really leaning pretty heavily on the Ablinks experience, and we were lucky enough to get Tony DeForolis, who is this ex-CSO of Ablinks, to advise us on this project. So one of the nice things is that delivery of one of these nanobodies to the terminal alveoli largely depends on the device used. So modern mesh nebulizers actually have solved this problem pretty nicely. Their kind of size range is right in the 2 to 5 micron range, and you can get devices that tune those particles a little bit larger or smaller depending on exactly where you want the deposition to be. For this application, it might actually be nicer to have almost a varied size range, so things both get deep into the lungs, but also perhaps coat the bronchial airways, and perhaps even some of the upper airway. So again, we're largely leaning on this experience from Ablinks and their ability to show that their molecule was effective in another respiratory virus in infants. And there's a lot of data about exactly what size of nebulized aerosol you need to get to these various regions.
Yeah. There's a large science around that. Is there anything about the overall charge of the nanobodies that is favorable / unfavorable for ultimately being successfully delivered?
I don't know the answer to that. There may be. We've mostly just thought about this from a stability perspective. If it's hopefully stable enough to be aerosolized, and that's what we're pretty confident about, that a reasonably large fraction of it could make it that deep in the lungs.
Thanks.
Aashish, that was amazing. I'm going to jump in here. I've got a question that builds on Brian's question. Are you guys looking into pegylation? Because some nanobody candidates are being pegylated, seems to improve pulmonary distribution. So yeah.
Yeah. So we haven't started working on any of that chemistry. And I think part of the challenge is any new kind of chemistry for one of these approaches raises the bar for what the FDA might want from a safety perspective, or what might be required to really understand the PK of what you're trying to work with. So a reasonably large fraction. Let me just kind of go back to this data from Ablinks, because it's pretty amazing, what they had worked on for their molecule. So this study that I just have in a little table on the bottom right here is a pretty amazing study. This is dosing this molecule, a trimeric nanobody, very analogous to the molecule that we've developed for SARS-CoV-2 in healthy human adults, and even simply knowing that the serum PK has a half-life of 20 hours, if you change even little things about the molecule, that's going to change. And I would venture that even if you were to go to like a single, monomeric nanobody versus a trimeric nanobody, this number will probably change. So I think, at least for us, the logic has been to try to stick as closely as possible to this overall architecture, because this data is relatively well known and probably applies to a molecule with a similar architecture.
Yeah, pegylation typically makes the half-lives longer, which is, a big reason why people do it.
Totally. Yeah.
But, and hey, I've got a general question for you and everyone wanting to deliver nanobodies as drugs. What about the loss of antibody effector function? How big a deal? I'm sure it's disease and condition dependent. For SARS-CoV-2, what is your impression of losing effector function and how that's going to play out?
Yeah, this is definitely so the effector functions are obviously probably quite important for some of the ways that the monoclonals that are moving forward work, but they come with the disadvantage that the IgGs really don't get into the epithelia very well, right? So on the one hand, they buy you probably important effector functions for viral control, but the flip side is that in the cases where people have looked at the amount of circulating IgG that gets into the lung epithelium, it's about 0.5% of the C-max dose, not even of of the circulating dose. So the challenge for aerosolized approaches is that the FC region is really the part that's somewhat liable and denatures when you try to aerosolize. So it's we think that simply blocking, at least for this respiratory virus, because that interaction is relatively well understood, that blocking that interaction is probably the main benefit, and the FC part is probably added benefit on top. And clearly for, RSV, simply blocking that fusion step is sufficient to even prevent replication in a pretty sick patient already. For other nanobodies, for other diseases or for other indications, I think it's a similar kind of balance. The FC for most antibodies buys you really long half-lives that you can't quite get without. But, and in most cases, one probably wants the super long half-life and potential effector function. And there's now good ways of engineering effector functions, out if you don't want them, but retaining some of the half-life extension properties. So I think, for drug development using nanobodies, really, I think delivery through the airways is probably a very uniquely privileged place to be, because that's something that regular antibodies can't do. But I think for most, drugs that you might want to deliver through the blood, I'm not convinced there's a whole large benefit for going the nanobody approach compared to a conventional antibody. There probably are some, but I don't think that's as large as for something targeted through the respiratory airways.
Yeah, I mean, of course, effector function can sometimes be very bad. So, like with SARS-1 had pretty big T helper 2 activation and eosinophil recruitment that created the lung damage, and certainly would rule out any antibody dependent enhancement. So, yeah, could be nice and tidy to not have it.
Sure, totally, yep.
Hey, I've got a really technical question.
Sure.
It went by quickly. I didn't quite catch. So you showed that when you matured nanobody 6, that you showed us a structure showing that the loops, yes, that. So, okay, so it is. Okay, so it is between, right, free and bound. So my question is, if you do have such a big conformational rearrangement upon binding, why aren't you paying, some sort of massive entropic tax that would make the, binding super weak?
Yeah, this is a great question and something we are totally scratching our heads on. Most of the time, maybe low affinity binders, micromolar affinity binders might have this kind of behavior where, that entropic tax basically is why it's a micromolar affinity binder. And as you affinity mature, you lock down these alternative conformations, and that's what drives a lot of the affinity gain. But this is a 500, picomolar binder, so not a lousy binder. And we are quite confused why, it's, why the loop rearrangements are so large. Now, one of the caveats of the crystal structure is that there are some lattice contacts, but even with that, I think it's pretty remarkable that the conformations are so different. So that's something we're trying to learn about our synthetic libraries. This isn't the first time we've seen something like this. We've gotten crystal structures or other structures of nanobodies out of our synthetic library. We often see that binding leads to relatively large loop conformational rearrangements, and that may just reflect something to do with the kinds of binders that come out of these synthetic libraries. And maybe, if we can control this kind of behavior better, maybe, do other kinds of maturation campaigns where we really lock down the loops, then, this 500 picomolar binder would actually be a 1 picomolar binder or something like that.
Yeah, okay, I've got one more sort of technical question, and then I think that the big question that's on everyone's mind. You may be the wrong person to ask about this, but I've been wondering if SARS-CoV-2 is using co-receptors as well, because in the decades after SARS-1 and MERS, co-receptors did emerge, and I'm seeing SLC6A19 kicked around as a putative co-receptor for SARS-CoV-2. Have you guys stumbled upon?
We have tried the various ones that have been proposed, not SLC6A19, but some of the others that have been proposed just biochemically. And this is again using purified prefusion spikes. That's not, it's not at least biochemically, we haven't been able to recapitulate some of those interactions. But I think even if there is a co-receptor, clearly this binary engagement with ACE2 is like one of the critical, if not the most critical aspect of this. So
Yeah.
I think the co-receptor stuff probably, if we can really pin that down, is maybe important for tropism or other kind of phenomena on top. But the very simplistic version of like, hey, this thing sticks to this other thing, and if you block that, it largely blocks entry, I think largely seems to be true for from the perspective of these kinds of strategies.
Absolutely. And okay, and then we're all dying to know, like, are you talking to industry partners about like making a lot of this stuff and getting into clinical trials? And if so, how's it looking?
Yeah, we're doing a lot of that right now. So we have a number of groups that are doing pretty deep dives, doing relatively, their appropriate diligence. And I think the big question from the perspective of, an industry group to take this forward at this point is, to what extent is an approach like this going to be available before some of the vaccines? And in a world where there are vaccines that are effective or not effective, or what is the probability of the timeline for vaccines? So I think all of these considerations are what need to be worked through. Our opinion is that even with vaccines, let's say available by the middle of next summer, which is kind of my bet on when they may be broadly available, an approach like this could provide added protection for people that are really high risk. So the vaccines are only going to be powered, at least some of those studies are only powered for 50% efficacy. So there's going to be patients that perhaps are so high risk that you really want a secondary approach. And that's again presuming that those vaccines are safe and we're on timeline for next summer.
Right.
So there are a number of groups that are doing their deep dives, and we're hoping that one of them pulls through and really has the resources and willpower really to push this forward.
Yeah. It strikes me that it would be very foolish not to move forward with another arrow in the quiver, and that, I mean, as far as I can tell, there seems to be a lot of money to put into development of these things, but I don't know.
Aashish, what are your thoughts on this being a prophylactic approach? For instance, could we just hand over an inhaler each to our healthcare workers or workers and the emergency or the front line, so that as a prophylactic approach, even assuming the vaccine's out there, do you think this could be an additional line of defense?
Yeah, so I'm slowly getting educated on what the challenges of actually thinking about how to design a trial for various things are. So the pipe dream for an approach like this is like a truly broad scale prophylactic that we hand to everyone, right? Like you want to get on an airplane now, like here, like it's an AERONAB, and you now like maybe we'll get less sick. So the challenge with really learning something like that, whether it's effective, you can imagine, is really quite the amount of money required to do a trial like that, get enough power to get a trial like that, and then the level of safety you have to have. All of that is really quite challenging for like that. By contrast, if you take people that are very early in the course of infection, or take household contacts where you think the probability of people in the household getting sick is pretty high anyway, then those trials are probably far easier to run and much more likely to show benefit, and the safety profile that you have to have is very different. So we think that very early applications of this would be for, again, patients that are very early in the course of infection and probably post-exposure prophylaxis. So people who have been contact traced and are waiting, like basically testing every day and are trying to figure out if they're going to get sick or not. And then more long-term, I think, running trials in patients that are high risk, like you mentioned, Sarada, healthcare workers or nursing home residents, or people in industries like meatpacking plant workers that are at high risk. That would be kind of the next line of potential utility.
Interesting. And I guess one last question before we let you go. You talked about aerosol, which, I mean, an inhaler is a very appealing way to deliver this. But is there any other mode of delivery that you guys are exploring?
Yeah, the other thing that we had been thinking about is intranasal delivery with one of these, just like a little simple spray bottle. The intranasal delivery is not trivial. It is a very, very, very complex, mostly because the clearance of, the nasal passageways is on the order of about 20 minutes, is constantly just, moving stuff out from the nasal passageways and into the oropharynx to swallow it or get rid of it. So there are a number of groups that have developed, ways of getting stuff to stick in that space, but really having it delivered, throughout the nasal passageways is a pretty challenging approach. So we're thinking about it. We're talking to people that have expertise in that area, but just out of the conversations we've had, it seems like it'll be pretty challenging.
Yeah, Ashish, I'll say again, that was really amazing, man. And this gives me a lot of hope.
Great, well, thank you.
Yeah, thanks for the talk and thanks for everything you're doing.
Great, thanks for having me.
Thanks for taking time, Aashish.
All right.
Bye, everyone.
Take care.